However, if that were just the case, then I can stop here and not have to talk anymore, not have to do any work. But what is basically unknown is what happens when we really pump up the data set size. Most of the work has been done with relatively small data sets, p less than 10,000, so it's really unclear what occurs with these larger data sets. And it really, at this point, the driving motivation was, well, are these default values, are they still appropriate? So it was basically at this point that I jumped into a project where I was handed a GWA data set and I was told, apply random forest to it and see what happens. So I'm going to walk through basically an investigation to figure out what is the best way to apply random forest to this data set, or this type of data structure. And while we talk about a very specific data structure, I think there are a lot of lessons that are generalizable to many other different types of data sets. So the data set used was a multiple sclerosis data set. MS is an autoimmune disease with a fairly well-documented genetic component. And it's well known that the HLA region of chromosome 6 is very strongly associated with MS. Oftentimes you'll see in GWAS studies, you'll see p-values of 10 to the minus 70. So essentially, there are very strong associations. And a 2007 multi-stage study was able to identify 16 SNPs across 13 different genes as also being associated with MS. Since that study, some of those SNPs have been further replicated, though some have not, indicating that there really is more work to be done. So the goals of this project handed me was, one, from an epidemiological perspective, can we identify novel SNPs or regions? And from a methodological perspective is, can we go about determining how random forest can be used for such data? And it's this second component that I'm going to focus on here now.
Did you try and get a handle on how much of the genetic variance those 16 SNPs would explain?
No, I did not. No. But for example,
people estimated it?
there is, not to my knowledge, but if you look at the HLA region, for example, just by using a few HLA SNPs, you can get down to about a 34% error rate. Just with a few HLA SNPs. So indicating that there is a significant amount in this HLA region, but there is more around there after that can be explained. But I don't know specifically about those 13.
How many of the 16 are in the HLA?
No, none of them are in the HLA. These are independent of the HLA, yeah.
But HLA SNPs were not among the most significant?
No. The goal of this 2007 study was to find SNPs outside of the HLA. Yeah, so these are spread out across the genome. I think encompassing, the 13 genes were on, about 8 or 9 different chromosomes.
And even though it's problematic, it's a good idea to try and estimate the percentage of genetic variance HLA plus these 16.
Yeah, no, I don't know if the initial authors in that paper, if that's something that they did look at or not. Yes, MS here is a case control study, so MS, we do have phenotypic information about the types of MS, and some of the other work is going, in our group is looking at, are there genes that are predictive of different types or subtypes of MS. But as mentioned, with all these complex diseases, you're dealing with a lot of disease heterogeneity. So that is definitely a weakening aspect in these studies, that you will have heterogeneous phenotype. So in dealing with random forest, there's a couple different tuning parameters that we can consider. The first and most basic one is how many trees to grow. And this is really just, the question about this is really just a computational issue of how long do you want to let it run, because unlike boosting, it for the most part, it does not overfit the model. And the general recommended default is between 500 and 1,000. Probably most important parameter is this mtry value, how many variables to select per node. And this is what will essentially control the bias variance trade-off in the algorithm. If we select very few variables per node, if we're only looking at a few SNPs, a few markers at every node to search over, we'll end up with very low correlation between our trees, so a low variance predictor, but each of our individual trees will have a higher bias. Conversely, if we're looking at many variables per node, the correlation between our trees will increase, so our variance will go up, but the bias of an individual tree will go down. So essentially what we're looking for is some value of mtry that'll minimize the overall error rate. Since we're dealing with prediction, we can think about should we upweight or downweight certain one of the classes. And while it's generally it's accepted that we should grow trees to maximal depth, that's something we can also question as a tuning parameter. Should we maybe cut the tree short? In terms of dealing with the data, we do have also some options too. There's a lot of, as mentioned, there's a lot of correlation in the data. Do we want to do any type of LD pruning? And I'll talk more about that. Another growing area of interest is this issue of subsampling. As with a lot of these models, we end up what are referred to as sparse solutions, where not all the variables will be pulled into the model, and therefore will have a variable importance of 0. So we can think of doing a recursive method of recursively removing out those sparse variables, refitting the model, again removing those sparse variables, and recursively refitting, trying to zero in on a most interesting set of variables. And while this is actually something that I haven't seen really talk much about with these types of methods, but removing dominant data. Now that we're moving outside of marginal tests, but looking at conditional effects, it's possible that by keeping in this really strongly associated HLA data with these really strong effects, that it can be masking other weaker effects. Essentially, if we think about we're growing a CAR tree, these strong effects are always going to be at the top of the tree. So anything else we bring into the model will by nature be conditional on that initial effect. So can we actually improve the game by removing out these very strong effects? So the data used was actually taken from this 2007 study, about 900 cases and 2,400 controls. The missing data was imputed with the standard package Beagle, and the final data set had about 325,000 SNPs across 22 chromosomes. We excluded the X chromosome. And the version of RandomForests used was provided by Sulford Systems. They helped us in terms of optimizing the code to really fit this data set. And the guiding principle for choosing the different tuning parameters and the data configurations was that which minimized the out-of-bag error rate. And the variables were ranked based on the permutation-based variable importance. So just a quick schematic. So it started with determining the optimal values of mtry and ntree. Recursively removed out the sparse SNPs. Then I did a rerun removing this chromosome 6p region. I did tests at different levels of LD pruning. Again, did this removing out of sparse variables, and went through 3 different methods of trying to confirm the final results. So the first consideration was how many SNPs to select per node. So what we have here is going along the x-axis are the number of trees, the y-axis is the out-of-bag error rate, and each of the colored lines are different values of mtry, starting with an mtry of 1, 2 times the square root of p, which is around the recommended default value, 10% p, 50% p, and p. So I'm really trying to cover a wide range of different variable search over. And what's noted is that these 2 lower mtry values end up with much higher final out-of-bag error rates. But once we really ramp up how many variables per node we're searching over, and we get a much lower final error rate with it being minimized in the green line by this 10% p. For context, we can think of an mtry equal to p as essentially bagging carts. We're looking at every variable at every node, so we're growing a CAR tree just on bag data. But the fact that these 3 mtries all close to each other does still speak to some of the robustness properties of the algorithm, but at the same time, it does speak to the idea that we can't just use the default values out of context. Some other effects of the mtry was that there was a pretty strong impact on the final sparsity of the solution. The more variables we looked at a given node, the sparser the final solution. Because if we're looking at more variables at a node, we're allowing the strongest variables to speak, and we're not forcing weak variables into the model. Some other considerations were the number of trees, and essentially the strength of the variables in the data set determined how quickly the number of trees converged. For here, you see the areas are basically converging after 200, 250 trees. However, for example, when I removed out the 6p data set with this really strong effects, it took much longer for the model to converge. It eventually did level off, but it does take longer. And the size of the forest, too, has an expected impact on the final sparsity.